Hello, good evening, everyone. Is my screen visible to all of you? Good evening, Shraddha. Good evening, Soumya. Good evening, Sarang. Good evening, everyone. Good evening, Siddhesh. How are you? Is the screen visible to everyone? Okay, thank you, Shraddha. Okay, thank you, everyone. So, yesterday we discussed about what is bioinformatics. We discussed why bioinformatics was created and what are the some applications of bioinformatics in different fields. And we also understood the term data driven biology. So, today we will learn where all this biological data is stored, because scientists all over the world, they generate biological data. They perform experiments every day. They sequence DNA, RNA and proteins. They publish research papers. They identify disease causing mutations. So all of this, it generates a huge amount of biological data. So the question is that where does this biological data go after the scientist generate it? So the answer is biological databases. So what is a biological database? A biological database is an organized collection of biological information. So what is this biological information? The DNA sequences, the protein sequences. So a biological database, it stores all of this information. It can store protein, it can store RNA sequences, it can store structures, research papers, genome information, or any other research informations. So all of the information which is available, which is biological information, so it is stored in biological database. So without these databases, scientists would have no efficient way to store the biological information. So databases, they are majorly divided into two categories. First is primary database and second is secondary database. So firstly, we will talk about the primary databases. So primary database, it means that these databases, they store the original experimental data. So what is this original experimental data? Means if there is a scientist, suppose, and that scientist, he sequences a gene and he uploads it directly. So that's become the primary data. Okay, if a scientist is sequencing a gene and he upload that data, that experimental data, directly in the database, so that database is called as primary database. So these are some of the examples of the primary database: GenBank, EMBL, DDBJ. So these are called primary database. Okay, so don't worry, here we will learn all these databases one by one. So are we clear with the primary database definition? Okay, so second type of database we have is secondary database. So these secondary databases, they contain the analyzed information. Now what do we understand by analyzed information? Means these databases here, they store the analyzed information which is derived from the primary database. So now we understand that primary database, they store the original experimental data. So if someone analyze this original experimental data, so they will have more information, means they analyzed this data. So that information is stored in secondary databases. So these are some of the examples of secondary databases. So are we clear with primary and secondary databases that majorly there are two kind of databases? One database which stores the original information and another database means secondary database that contain the analyzed information. So majorly these are the two types of databases, but we have one more type of database which is known as composite databases. Okay. So these databases, they combine the information from multiple sources. That is why they are called as composite databases. So these are the three type of databases, and this example of composite database is UniProt. So we will learn about UniProt as well. So are we clear with databases, different kind of databases, that they are divided into three categories? So we will learn primary, secondary and composite databases one by one. But firstly, we will learn one of the most important platforms in bioinformatics, that is NCBI. So first of all, let's open our Google because we are going to open the NCBI. So here, this is Google. In the search bar, we are going to type NCBI and press enter. So this is the official website of NCBI. So NCBI, it is known as National Center for Biotechnology Information, and in short, it is called NCBI. So NCBI here, it is part of National Library of Medicine. It is part of National Institute of Health, and it is also part of the USA government. Okay. So almost every researcher uses NCBI because as NCBI was created to store and organize the biological information. So let's click here and let's open NCBI. So here, this is the homepage of the NCBI. And NCBI is very, very important because before NCBI, the biological data was scattered everywhere. So NCBI solved this problem by creating centralized databases. Now, what do we understand by the term centralized databases? So look at here, look at the cursor, you can see a drop down menu here. Here it is written all databases. So this is a drop down menu. So let's click here. So these are all the centralized databases. Means we can select any database from here. So today almost every bioinformatician, every genetist, every molecular biologist, if we talk about drug discovery scientist and every NGS researcher, so everyone uses NCBI. So for example, here we have gene, we have genome database. All of these are databases. Okay. Here we have protein database. Suppose I want to select the protein database. So we will just simply click on it. So we have selected the protein database. Okay. Now the most important thing to understand here that NCBI, it is a huge platform. Okay, NCBI is a huge platform containing many databases and many tools. So NCBI is not just one database. It is a huge platform for many other databases. As here we can see, these are the centralized databases. Means NCBI has created centralized databases. So we can just select it from here. For example, I want to select gene database. So I will just select this. Okay, so NCBI has made it very easy for us. So similarly, look at here on this left hand side, we can see many different categories. We can see all resources, we can see DNA and RNA, gene, genetics, homology, proteins. So these are different categories here. So these categories, they are basically the division of biological information. Now, what do we understand division of biological information? Because NCBI contains huge amount of biological data. As we can already understand that there are many databases. So these databases, they contain many information, right? So NCBI itself contains huge amount of biological data. So to organize everything properly, NCBI separates the data into different categories here. For example, here we have DNA and RNA section. So let's click on here, DNA and RNA section. So here we are seeing different tabs. We are seeing databases, we are seeing download tab, submission, tools, and how to. So here in this database tab, so every database that you are seeing here, assembly, biocollection, bioproject, here we have GenBank, here we have nucleotide database. So these are all the databases related to DNA and RNA. Means these databases contain the information only related to DNA and RNA. So if we talk about protein, so let's select this protein category. Let's click databases. So here we can see we have protein clusters, we have protein database. So here these database only contains the information related to proteins. Okay, so are we clear that different databases contain different information, right? So here next we have this downloads tab. So here in this download section, it helps the researcher to download the biological data. So if we want to download any kind of biological data, we can use this download section here. Then here we have this submission section. So as you can see, if you select any category, suppose you select proteins here again. So these tabs are the same. If you select, let's select homology. So again, these tabs are the same. Let's select chemicals and bioassays. So these tabs are the same in every category. Okay, so that means here, if we click on downloads, we have different option because this category contains different information. Here we have chemical and bioassays, means this category here, it stores chemicals. It may store drugs. Okay, it may stores molecules. So if we want to download a particular type of biological data, we have to select these categories and here we have to go to these downloads. Similarly, in the submission section here, if this section here, it is used when scientists want to upload their own data. Now, for example, there is one scientist and that scientist is working on a particular protein. Okay, and that scientist want to submit his data on the NCBI. Now, he cannot just click on any random categories here because he is working on protein. So, he will have to select the protein category and submissions here to upload his protein work. Okay, means protein information. So we are talking about different categories because these different categories stores different kind of biological information. And similarly, in the tools section here, we have different tools according to these different biological data. So for example, here in the proteins, we have BLAST, here we have CN3D. So these are the different tools, but they are only related to protein. Similarly, if I click on chemicals and bioassays and in this tool section, so we can see the tools has been changed. Okay, so these tools are only used for these chemicals and bioassays. And then we have the how to section. So this section, it helps you how to use this. So it contains all the tutorials and all the help guides. Okay, for example, let's click on data and software and you want to see how the things work, how to submit the data to the NCBI. Okay, so here you will see all the help guide here. Now, for example, if you want to click on protein category and how to section, so here how to submit the protein data to the NCBI. Okay, and these are all the help guides. So again, all these tabs are same in these different categories, but these different categories are related to different kind of biological data. So do we have any question here? Yes, Brendan, there are reference genes here as well. So do we have any question related to these categories? Do we understand different databases contains different biological data and all of the data is divided here in these categories? Do we have any question? So let's go back to NCBI home. Now, one more thing, very important thing to understand is that we just talked about DNA, means RNA, protein sequences, chemicals. So here we are talking about sequences, right? Now, NCBI is not only for biological sequences. NCBI also stores the scientific knowledge related to biology and medicine. For example, researchers, they constantly publish the research papers. They constantly publish review articles, the clinical studies, the disease reports. There are some scientific textbook as well. So here we have this literature section. So this literature section here, it helps the researcher access all the scientific information. So let's click on this literature category and let's click on the databases. So here, these are all the databases that contains the literature, means the written work, the scientific work. Okay, if we talk about articles, if we talk about research papers, if we talk about some books, so all of these databases contains these data. Okay, so here you can see, first of all, the name of the databases is bookshelf itself. So from the name itself, we can have an idea that this database contains books. Okay, so here in this literature category here, we can find all the databases from where we can access the scientific paper or any literature work or any research papers. Okay, so for example, let's see PubMed here. Here we have this PubMed database. All of these are databases. Okay, so here we have this PubMed database. So this database here, it is used to search the research papers. Means if you want to search for any specific research paper, then you will use this PubMed database. Similarly, we have PubMed Central. So the name is similar, but their work is actually very different. Okay, so in this PubMed database, means if we want to search for specific research papers, we will use this PubMed database. Then we have this PubMed Central. In short, it is called PMC. So this database here, it is used if we want to read the complete full text research articles. Okay, so there is a difference between these two. So we will learn about these two in today's lecture itself. So let's come back to the bookshelf again. Again, we just understood that the database name is bookshelf, means this database contains the scientific books or it contains the educational material. Similarly, we have MESH database. So this database here, it contains the medical terminology or it may contain the scientific topics. Okay, so here we have description as well. We can read this description if we want to learn about some specific databases. Now, very important thing to understand here that you do not need to memorize every category right now. Okay, there is no need to learn everything here. Right now, our main goal is to understand one thing, that NCBI is not a single database. NCBI is a complete bioinformatics platform which contains many databases and there are many analysis tools. And here, these categories, they help here to organize all those resources properly. So let's click on DNA and RNA category one more time. And here, let's see tools. So we talked about tools as well, correct? So in DNA and RNA, we use this BLAST tool. And this BLAST tool is also used in proteins as well. And we also saw that in protein category, in the tool section, we have this BLAST. But again, if we talk about chemical and bioassay category, so there is no BLAST, right? So for different biological databases, there are different tools. And NCBI, it analyzes, it organizes these tools very efficiently here. So are we clear about these categories, all these databases? Did we have any idea or do we have any confusion? So did we understood that NCBI is not just a single database, it is a huge platform and it contains many tools, it contains many information. Now look at here, on this right hand side here, we have popular resources. So here the name is popular resources because these resources, they are very frequently used among researchers. Okay, these tools, here we have these databases, here we have this tool, again this database. So these are very frequently used by researchers. That is why we have this particular section here, popular resources. So first of all, let's talk about PubMed. So this is the PubMed, this is the database, and we understood that this database is used for research papers. So again, NCBI is not only for sequences, it also contains the scientific literature. So let's click on PubMed. So this is the PubMed here, okay? And here we can search for anything. For example, I want to search for breast cancer. So let's select the breast cancer and let's click on search. So here we have different search results. Now what are we going to do? We are going to observe. Okay, now observe here very carefully. So I'm going to click on this first search result. Now this is the title of this article, breast cancer presentation, investigation and management, whatever it is. Now look at here. The first thing that we have is abstract. Second thing we have is similar articles. Third thing we have this cited by, and here at the last we have publication types. Okay. Now what I'm going to do, I'm going to click on this second search result. So let's click here. So here again we have the abstract, again we have the similar article, again we have the cited by, again we have the publication types. Now let's do this one more time. Okay, so let's click this third search result. So here again we have abstract, here we have conflict of interest statements, again we have the similar articles, cited by, and here we have references. Now this is something new, again we have the publication types. Now did you see something similarity? Did you observe something? Let me know what did you observe in this PubMed search result. Let me know in the chat box what did you observe. Anyone? What did you observe? We saw these three search results and there was some similarity here in these three search results. So did you observe anything? Anyone wants to answer? Okay, so in these three search results, what we saw? Yes, Akshari, very good. A format is followed. But here, did you see, we searched for breast cancer, means in this PubMed, we want to search for a research article, correct? So if I click here, so can I see the complete research article here? I have the abstract here, I have the similar articles, I have this cited by section here. So here this is the end. So did I see here the article? Did I see any article here? Right. Very good, Akshari. Very good. We did not see the article here. So that is what the PubMed is about. PubMed is only used when we want to search for the articles. So here what we observe that in these three search results, it contains the paper title, it contains the abstract, it contains the general information, and it contains the publication details. So PubMed here, it mainly helps the researcher to find the scientific papers. Okay. So one important thing to understand about PubMed is that PubMed does not always contain the complete research article. It only provides the paper information, the abstract, or a link to the publisher website. So where do we read the complete full text research article? That is where we use the PubMed Central. So this database, PubMed Central, in short we call it PMC. So here this is the database that stores the complete full text research articles. Okay, so let's click here. So here let's again search for breast cancer. So let's select this breast cancer here. Let's search in PMC. Now here we have these search results. So let's click on the first search result. Now, what do we have on this page? We have the abstract. We have the abstract here. Then we have the introduction. Then we have the case description here. We have some figures here as well. Here we have the discussion. So can we say that this is an article? Right? It is very different from PubMed. So do we understand the difference between PubMed and PubMed Central? That PubMed helps you search for the research papers, but PMC, it helps you read the full research articles. So let's click on second search result. Okay, so this is the title of the paper. Let's scroll down. So here you can see we have many things in this article. We have abstract here, then we have the background. Here let's go to discussion. Then we have limitations, and at the end we have this associated data. So this is what we call article, right? So do we understand the difference between PubMed and PubMed Central? And do you understand that when you want to use PubMed and when you want to use PubMed Central? Okay, very good. Now let's go back to NCBI. Okay, so now we have this bookshelf database. So let's click on this bookshelf database. Now, this bookshelf database, so as you can see here, we had all the centralized databases. So here we selected the books from here. So NCBI did it for ourselves, right? Because we clicked on the bookshelf database. So let's click here again. So here we have the books. So this bookshelf database, it mainly stores the scientific books, it stores the textbook, it can sometimes stores the review articles as well, some manuals, or mainly educational resources. Okay, so again, instead of storing only biological sequences, NCBI has bookshelf and it stores the scientific knowledge and some learning material. So let's search here bioinformatics. So let's select bioinformatics, click on search. Now here, what are you seeing? Can we say that these looks like books? Okay, and these are all the open resources. So here I searched for bioinformatics in the search bar, and here we are seeing that the bookshelf database, it provides us all the educational resources. These are all the scientific books, the learning material for you. For example, let's click on neuroproteomics. So here we can see these are the chapters, chapter 10, chapter 7, chapter 6. So where is chapter 1? So here is chapter 1. So this is a book, right, in the database of bookshelf. So if you want to study a particular concept, what you can do, you can just search here your particular interest of the concept and you will find all the learning material here in these search results. So are we clear about the bookshelf database in the NCBI? Okay, very good. Now, next we have BLAST. Now, we talked here that PubMed, Bookshelf, PubMed Central, these are the databases. Now, BLAST here, it is a tool. Okay, BLAST is a tool which is used for sequence similarity searching. So, in our upcoming lessons, we will learn about BLAST in detail. Okay, so for now, just understand BLAST is a tool which is used for sequence similarity searching. Again, we have nucleotide database, we have genome database, we have SNP database. So what is SNP? SNP, it is actually a type of a variant. Do we understand variants? Okay, we understand mutations, right? We all have heard of mutations. And by mutation, one thing comes to our mind that there is some change in the DNA, right? So that's the same thing about variation. So here we can see that we have a category of variation as well. So in this variation, we have different types of variation. You can just uh, compare it with mutations. Okay, it is not different than mutation. The meaning is the same, that there is some difference in the DNA. Okay, so there are different types of mutation, and one type of mutation is SNP. So here in this database, you will find all the information related to the SNPs. Okay, again we have the gene database, we have the protein database, and here we have the PubChem database. So let's open this PubChem database. Now, what is this PubChem database? So this database here, PubChem, it is a chemical database inside the NCBI. So this database here, it stores the information about the chemicals, it stores the information about the drugs, some molecules, compounds. For example, if I search hemoglobin here, okay, I'm searching for hemoglobin. Now here we are seeing three different categories: compound, gene and taxonomy. So here we are not going to select anything from here. We are simply going to click on the search. Now I searched hemoglobin here inside PubChem, and what you can see here, you can observe something very interesting here. Here we are seeing different categories. We are seeing compounds. Let's click on substances. So we are seeing substances. We are seeing RefChem. Here we are seeing bioassays, genes, proteins, pathway, and literature is available here as well. We understand literature now means all the scientific work, all the scientific writing, research paper, articles. Okay, and here we have the patents. So researchers, they use PubChem when they want to study the chemical structure, when they want to study a particular biological activity, or if they want to study the drug related information, because PubChem here, it stores the information of the drugs, the molecules, the compounds. Okay, so here this PubChem, it helps organize all the information together. So this is why PubChem is extremely useful in drug discovery, because along with the compounds, you can search for substances. Along with the substances, you can search for genes, proteins, or if you want to see pathways, means how a particular function works. Okay, for example, there is a protein. How does that protein work? What is the pathway? So for example, here's hemoglobin. So let's click on pathways. So here we can see different of search results. Okay, and here we have literature, means if we want to search for some article or research paper, so we can search it in the literature section. So PubChem here has organized everything together. So do we understand PubChem, that when we have to use the PubChem database, when we are studying about any drug, when we are studying about any chemicals or any biological activity, so that is where we use the PubChem database. Now let's go back to the NCBI homepage. Now here in this section, we have the NCBI news and blogs, means all the news related to NCBI or all the NCBI blogs, you will see it here. Okay, for example, if we click it here, so here you can find all the news and the blogs of the NCBI. Okay. Now, this is the middle section of the NCBI, and in this section you are seeing some different things. You are seeing the section submit, download, learn. So what is these sections about? So first section is submit. So this section is used when the researchers want to upload their own biological data to the NCBI. Okay, so that is why NCBI stores many information, because researchers or scientists, they can directly upload their data here to the NCBI. So for example, in a lab, a scientist perform a sequencing of a new bacterial genome. Now they can submit their data directly to the NCBI. So can we divide NCBI to a primary or to a secondary database? Can we divide NCBI to a particular kind of database? Who will answer to this question? Because I just give you the example that if a scientist is working on a gene, he sequences that gene and he uploads the experimental data directly to the NCBI. Now the question is NCBI a primary or a secondary database? What is your answer, everyone? Okay, Induja, okay, Soumya Jyoti, but we just talked that NCBI contains a lot of databases, right? We just talked about that NCBI is a huge platform. NCBI is not just one database. That is why I asked you this question, that is NCBI a primary or secondary database? Okay, so NCBI is not just a database, it is a huge platform for other databases. Okay, so I want you to do your own research on this question. Is NCBI a primary or a secondary database? Are we clear with this? Okay, very good. Now the second section we have is the download section. So this section is used if we want to download a biological data from the NCBI to our computers. Okay, so that is where we use this download section. Now next section we have is the learn section. So this section, it contains the tutorials, all the help documents, means how to use NCBI. So this section here, it contains all the training materials. So next we have the develop section. So this section, it is actually mainly for the programmers and the bioinformatic developers. Okay, because this section here, it provides the APIs, it provides the coding libraries, the automation tools. So here, this section, it helps the developers build the bioinformatics pipelines and the softwares. Okay, so this is for programmers and the bioinformatics developers. Now, this next section we have is analyze. So this section here, it helps users to find the analysis tools. For example, let's click on this. So what are we seeing here? We are seeing tools, and this is the description of all these tools. So this section here, it may contain BLAST. Here we can see BLAST. It may contain the sequence alignment tools. It can contain the primer design tools, or it may contain some genome analysis tools. So all the tools are here, and here is the all the description of the tools. Okay. So actually this is where bioinformatics analysis happens, because we use bioinformatics tools, right? Now let's go back, and here we have this research section. So this section here, it contains all the NCBI research projects. Okay, it contains all the collaborations of the NCBI, all the scientific initiatives. So this area is more related to the NCBI's own scientific work and collaborations. So are we clear what we have learned till now? Do we have any confusion related to any concept? Are we clear what we have learned till now? Okay, so we just understood that NCBI is a huge platform. It contains many databases, it contains many tools, right? Inside the NCBI, we have gene database, we have protein database, we have genome information. So there is one question, and this question is very important, that if NCBI contains so many databases, then how does NCBI search all this information efficiently? Because here we are seeing this is a very efficient work. Whoever designed this website, whoever designed NCBI, the work is very efficient. So for example, if I want to search for hemoglobin, right, and there are different databases. There is a PubChem database where I can search for hemoglobin. Now if I want to search for research, then we have the literature here. Okay, so everything is organized here inside the NCBI, but the data is very vast. Data is very huge. So the question is that how does NCBI search all of this information efficiently? So the answer is that NCBI uses a system called ENTREZ. ENTREZ. It is called ENTREZ. So what is ENTREZ? ENTREZ is a data retrieval system used inside NCBI. So in simple words, this ENTREZ means this data retrieval system. It helps the users. We are the users, right? So this ENTREZ, it helps the users to retrieve and to connect the biological information across different NCBI databases. So we are going to see one example. For example, here if I search BRCA1, okay, I have searched for BRCA1. Now let's click on search button. Now what are we seeing here? Do I see literature here, which is a different section, different database? Do I see genes here? Again, different database. Proteins, different database. Genomes, clinical, PubChem. So these are all the different databases, and ENTREZ here. So we searched BRCA1. So all the information of BRCA1 which is present around all the databases, Entres has connected everything together here. Okay, so instead of manually searching for every database separately, we can just select from here. For example, if I want to see bookshelf, okay, for BRCA1 I want to see some learning material, some books, so here I can directly select books. For example, if I want to see the compounds, here I can just select on compound. So there's no need for me to manually open the pubchem, right? I can select anything from here. So do we understand that Entres is a data retrieval system in NCBI? And one more thing to learn about Entres is that sometimes people confuse the Entres with the database. Okay, Entres is not a database, it is a search, it is a data retrieval system which is used to access the NCBI databases. So whenever we type something in the search bar here, we are actually using the entries system. Okay, so are we clear with the entries? That entries here is connecting all these databases together. Are we clear with the entries? Okay, so there is one more important reason why entries is extremely very important, because right now what we did here, we are using the entrance manually. Okay, we just type BRCA1 and entrance is automatically used here by the NCBI because it is a data retrieval system, right? So right now we are using the entrance manually through the NCBI search bar. But later in bioinformatics, we often work with programming. Okay, you will work with programming later, and you will also work with automation as well. So for example, let's take an example that you want to download 100 sequences from the NCBI. Okay, you want to download 100 sequences from the NCBI. Now that will take a lot of time, right? Because we are talking about 100 sequences and you are downloading it manually one by one from the NCBI. So that will take up a lot of time, right? So instead of manually doing it, what we do in bioinformatics, we use programming languages like Python or R to communicate directly with the NCBI. Okay, so as you continue learning bioinformatics and programming, you will see entries again and again, because when you will do programming, so let's take one more example as well. For example, you know Python, you know the programming language Python, and through the Python, we all know what we do in Python, we write some codes, right? Now you want to download 1,000 sequences from the NCBI, and in Python you will write a particular code that you want to download 1,000 sequences from the NCBI. Now, instead of manually opening the NCBI, you are writing a code. And in that code, you will be using the entries system. Okay, so here we are just manually using the entries in the NCBI search bar. But in the programming, to build a connection from your code to the NCBI, you will use the entries system. Okay, so that's why entrance is very important. So understand entrance, that it is a data retrieval system. So are we clear again with entrance? Okay, very good. Now let me give you one more assignment. So this week you will have two assignments. Okay, so every week you will work with two assignments. So this is your assignment number 2 and you have to submit your assignment by Sunday. Okay, so in this assignment, what you will do, you will choose any one human disease of your interest. Okay, any disease that you are interested in, you will select that disease. For example, Alzheimer, diabetes, asthma, whatever disease that interests you, you can select that. Okay, and after choosing that disease, your task is to investigate that disease using the databases that we learned today. So let me show you the task that you will be performing. So in the first task is the disease itself, that you will select the disease and you will write some 2-3 lines about that disease, means what is that disease about. Okay, So this task is very simple. Then in the second task, what you will be doing, you will be using the NCBI gene database. For example, here, let's open the NCBI again. So here from this is the gene database. You will just simply select it. Okay. And here in this search bar, you will type the interest of your disease. Okay. So in the NCBI gene database, you will find one gene that is associated with your disease. Okay, you will collect the gene name, you will collect the organism, you will collect the chromosome number. So basically what you will be doing here, you will be using the gene database. Then in task 3, you will use nucleotide and protein database. In the same way that we selected gene in the NCBI, in the same way you will select the nucleotide and you will select the protein. So by using these two databases, you will find one DNA sequence and you will find one protein sequence related to that gene. Okay, so here in this database, you found one gene and from that gene, you will retrieve DNA sequence and protein sequence. Okay. And then in task 4, you will use the PubMed database. And in the PubMed database, you will find one recent research paper which is related to your disease. Okay, so you will collect the paper title, the journal name, the publication year, whatever the information is present. Okay, and then in the fifth task, you will use the bookshelf database, and in that you will search for your disease and you will find one educational resource. Means in that book, for example, if you select the Alzheimer disease, so in the bookshelf database you will find one particular thing which will be explaining the Alzheimer disease, but you will be using the bookshelf database. So are we clear with this bookshelf database? And then we have the task 6 means the submission. So what you will submit? So you will create a table, and the table, it will contain only two things: the database that you will be using and what information you find using that database. For example, the first database you use is gene. So here you will write all the information that you collected from the gene database. For example, the second database you used is PubMed. Okay, so from that you will write all the information, the third database you used, and then the fourth database you used. So you will submit this table. So are we clear with this submission that you have to submit this table, which includes all the database that you used, and here on the right side it will include all the information you retrieved from that particular database. So are we clear with the assignment? Okay, so these two assignments you will submit it by Sunday, and also I will recommend you that you must collaborate in the group because it will help you in better learning. Okay, because different people have different opinions, so you will get to know something, you will get to know the new people, some new things. Okay, so collaborate in your group. Okay. Okay, now everyone kindly raise your hand so that I can take the screenshot. Let me mark your attendance. Okay, very good everyone. Everyone is present. Okay, so I have taken the screenshot. Okay, so if you have any questions, you may ask. Otherwise, you can leave the class. Okay, thank you everyone for joining. Yes, Brandon, what do you want to ask? Brandon, that depends on your research, how much you want to study. Okay, I can just only guide you that this is the gene database. You will find all the information here. So it depends on you how much you want to study. Okay. Okay, everyone, have a good evening.